Okay, here's a little 3-slide thing, and we're going to cover the rest of the proteins. So one of the questions is going to be, tell me what each of the proteins does in poliovirus. So we already covered the P1 region, which had VP1, VP2, VP3, which together form the capsid, VP1 being the five-fold axis that actually binds the receptors and has the fusion peptide and makes the little hole in the cell. VP4 being like a door or like a little gatekeeper that's going to escort along with the tails of VP1. They're going to escort the VPG right into the cytoplasm. And VP2 and 3 just being dumb bricks that help build the wall. So then the P2 region is where things get really interesting. So you've got your 2A, which is the protease, and this is the one that cleaves the EIF4G, which is like a bridge protein that's needed to begin cap-dependent translation. And so once we cleave that, we cannot translate anything with a cap anymore. We can only translate things that have an IRES on them. Yeah. Now, 2A also has another function, which is especially dastardly. It destroys nuclear pores. So we've seen a couple viruses now, the flu virus and HIV, that were really careful about making sure they hold open the door of the nuclear pore. Picornavirus doesn't really need the nucleus. It can actually grow in a cell when you take the nucleus out. And so what it does is it just blows up the door so that there is no way for the cell to get any more messenger RNA out. So even if the cell tries to make some kind of immune response, there is just nothing it can do, because making an immune response always involves translating some sort of new protein, which generally means it's going to come out of the nucleus. So yeah, that's a nice way that picornaviruses really beat up on the cell. All right, then you get 2B and 2C. Now they start out as a polyprotein, like a miniature polyprotein of 2B and C that lasts a long time. And 2B and C together forms these things. It makes lipid membrane, it causes the cell to make lipid membrane, so it fills up with these little bubbles, and it prevents protein secretion. So it's doing something to the Golgi, a little bit harmful to the Golgi. The little lipid membranes are going to form, this is where the viral proteins are going to form up, so it acts as like a little anchor that holds the viral polymerase down so that the polymerase can form up little virus factories, just like that. Okay, and then eventually the 2BC unit, and we're going to need to know that as part of the question answer. The 2BC unit gets cut into 2B and 2C, and it's going to be cleaved by the 3C protease, and we're going to get to that in a minute. 2B all by itself is a viroporin, which means a thing that forms pores in cellular membranes, just little tiny holes. This is going to be very similar to the ion channel M2 for influenza and to the ion channel E protein for coronavirus. But since this virus is not one that is going to carefully bud from the membranes, it doesn't even have a membrane, this has a different function. We think this is just to mess with the insides of the cell and kind of disable it. So the cell will store up all kinds of ions in different chambers. Like it'll store a lot of calcium in the endoplasmic reticulum sometimes. So it can release it at different times. This is part of how muscle cells work. And the viroporin just kind of pokes holes in things and lets all these ions just move around and go basically wherever. So it's just a giant jerk move and a bit of a middle finger from the virus to the cell. Now 2C is a really interesting one. So 2C looks just like a helicase structurally, but it does not function like a helicase. So it can use ATP, but it does not unwind RNA when it does that. Doesn't seem to have that sort of helicase activity, but seems to be descended from a helicase. And so what we think this may do is either it's another membrane anchor for viral RNA synthesis, or more likely, we think this could be the loader, the protein that loads the RNA into an empty capsid once it's made, and then just sort of kisses it goodbye and sends it off to go infect another cell, cause a little more polio in the world. Okay, so that was the P2 region. Here's the P3 region. So P3A has an AB, and the the 3AB protein, which is a polyprotein, is going to be bound to the genome during RNA synthesis. So it's not actually just the VPG. That's the 3B region. 3B is VPG. 3B VPG. And you're going to want to make little flashcards or something like that to study this. Make sure you get, or just a nice little chart that you can study so that you know, 3A. Oh, yeah, I know what that is. And so 3A is a very small protein, and it absolutely blows up the Golgi apparatus. At least the 3A of poliovirus does this. And so by blowing up the Golgi apparatus, 3A can effectively stop any signaling to the outside, because signaling is going to come in the form of translating new proteins and then exporting those new proteins, little proteins like interferon. And so if we've blown up the Golgi apparatus, that's like the central bus station for all transport in the cell. And if you blow it up, there's essentially no transport left. So it's another little thing. The virus says, well, I don't need this, so let's just destroy it. Yeah. Make life simpler, right? Okay, so 3B is going to be the primer for RNA synthesis, and it's going to be attached to every viral genome. But while we are actually synthesizing it, we synthesize it with 3A and B together. And then once we've started synthesizing, then we're going to cut 3A apart from 3B. And that's going to be that 3C protease that does the main thing. So then 3C. All right, so 3C is a protease. 3D is a polymerase. But the thing is, when they're together as 3CD, it makes the protease better. So 3CD is a super active version of the protease. That's why I drew big scissors there. Yeah, look at that. And so this is the one that's going to go along and just cut everywhere, all up and down the P2 region and the P3 region and a little bit in the P1 region, and free up all those proteins. And then once one copy of 3CD gets cut by another copy of 3CD, then you will have just the 3C and the 3D. Now 3C protease on its own is pretty weak sauce. It doesn't really do a good job of as a protease anymore. That's kind of how the off switch. That's how the virus turns that off. 3D, though, is a polymerase, so this is going to copy the RNA, and 3D does a terrible job being a polymerase while it's attached to 3C, but as soon as you've got 3D on its own, then boom, it takes off, and it is going to make primers and copy the positive and negative stranded RNA. So VPG actually serves as the primer for RNA synthesis. And so 3D polymerase comes along and it will add two U nucleotides, two uridines, to 3B. And so then you'll have VPG and then a phosphate and then a uridine and then a phosphate and another uridine. So they call it VPG PU. That is the actual primer that we're going to use to make positive strands and negative strands. And so, they both have a VPG at the end, as far as we know. And the reason they have it there is because the polymerase can add nucleotides to VPG, but it can't just start making nucleotides on its own. It just has this one special power, and that's how it gets its primer. The polymerase makes the primer out of VPG. And so that's why VPG is on the genome, because it started out the genome. It was the primer for the genome. So that's kind of cool. Okay, so here's a nice little overview of the same thing. So here we've got our 5' untranslated region. This is all of our IRES structure, the internal ribosome entry site. There's our VPG at the beginning. There's a couple other sequences that aren't involved in binding the ribosome directly up here, and these are going to bind directly to the 3D polymerase, so it can recognize that. Down here is the poly A tail, and in the middle is we're going to have our genes, and that CRE, that cis-acting replication element. There we go, I can say it, is going to be in the middle of this. So it's going to be somewhere in here, and it moves around on different viruses, but pretty close to the middle. We're going to translate this one big gene to make one big protein, which is called a polyprotein, because it's lots of little proteins all joined together. Then we're going to process that into 3 regions, the P1, P2, P3 regions, and then we're going to keep on processing that. And by processing, I mean cut with the 3C protease and the 2A protease and the cellular protease right up here between VP2 and VP4. And yeah, that's basically what's going on here at the bottom. These are nice little descriptions, very short descriptions. I have longer descriptions, and I tell you what some of the pieces do that are joined together up there.